Okay guys, let's continue the notes. Alright, we're going to flip to the next page. Alright, and we're going to talk about active transport next. Okay, active transport requires energy and a protein. So that protein uses energy to move a molecule from a low concentration to a high concentration, which is the opposite of what it wants to do naturally. So active transport is when a transport protein sometimes called a carrier protein requires energy to help move particles across a membrane against the concentration gradient. What I mean by concentration gradient, I mean from low concentration to high concentration. So, the example that I used in class today, a few years ago I went on a school trip to the zoo, and we took 4 buses, and when we went to leave, we loaded everybody on the bus, and bus number 4 did not start. So, in order for us to get all of the kids home on time, we had to take all of the students on the broken bus and put them on the other buses. Okay, so that meant it took a lot of energy to convince high school students that they needed to sit 3 to a seat. Okay, we needed to have the buses at maximum capacity in order to get everyone home on time so that they could catch the bus from school to home. So it took a lot of energy to move those students from low concentration on 4 buses to a higher concentration where they were all on 3 buses. So that took energy. That was active transport. Now we're going to talk about the 2 processes that cells use to either get matter into the cell in the form of large particles, or how they get rid of particles. So the 2 processes are called endocytosis and exocytosis. Endocytosis is when the cell is bringing matter into the cell. It looks like the word enter, so I always think enter. Things are entering the cell. So this is when large particles are engulfed, then enclosed. I spelled enclosed wrong, guys. Sorry about that. Are enclosed by the cell membrane and moved into the cell. Alright, so say that this purple dot is the substance that I want to engulf. I want to bring into my cell. So my cell will actually wrap the membrane around it so that it looks like this. Alright, and eventually the membrane over here will meet the membrane over here, and the plasma membrane will surround this, and this just became a vesicle. This became a substance covered with a plasma membrane and brought into the cell. Eventually, that cell membrane will break down, and this substance, this purple dot, will be used by the cell. Our next process is the exact opposite of that. Okay, it's called exocytosis. I always think of exo. It looks like exit, so things are leaving the cell. They are exiting. So this is when large particles are released from the cell by secreting or expelling them. Alright, so my cell has this purple dot that it wants to get rid of. Alright, and so the cell is still around it, but the membrane begins to pinch in around it until eventually it closes in and it sends this guy off out of the cell. So that is exocytosis. Alright, now we're going to move on to the chart that looks like this, that says the effect of osmosis on cells in different solutions. Now, I made a slight typo on mine, so whenever I upload this document to Classroom, I will fix this. This should say hypotonic. So, before I upload this document to Classroom, I will fix that so that it actually says it on yours. So, before we get started, I am going to define what's going to happen in each of these, and then it will be your job to either print this out and write them in, or type it directly on the document and then find pictures from the internet to fit in here, or draw your own. That is completely up to you. Okay, I am simply going to define them here for you. Then it is your job to find pictures online of what these look like and paste them in or draw them. So in a hypertonic solution, the solution has a higher concentration of dissolved substances. Solution has a higher solute concentration. Which means dissolved substances. Solution has a higher solute concentration. That is the stuff that is dissolved. So the cell begins to shrink. So the solution has a higher solute concentration than the cell. Causing the cell to shrink. And that is in an animal cell. In a plant cell, the vacuole, the central vacuole that holds the water, will collapse. Alright, so the central vacuole will collapse. In an isotonic solution, the cell neither gains nor loses water. Water enters and leaves the cell at the same rate. Because the solute concentration is the same inside and outside the cell. Inside and outside the cell. So when one water molecule moves in, one moves out. There's constantly the same amount of water going in as going out. And lastly, hypotonic. Okay, this is when the solution outside the cell has a lower solute concentration. Remember, that is the stuff that is dissolved in the water. A lower solute concentration than the cell, causing causing water to move Wow, I'm struggling writing today. Causing water to move in to the cell. In an animal cell, the cell will burst. Because too much water has flowed into the cell. In a plant cell, the central vacuole will swell. So a good rule of thumb to remember with this is that salt sucks. Salt will suck water. It'll draw water to it in whichever way there is more dissolved solution. There's more stuff dissolved in that water. So wherever the salt concentration is higher, the water will move in that direction. So what I want you guys to do now is either go online and copy and paste a photo of each of these scenarios into the appropriate boxes, or find a picture online and draw it as best you can. If you have any questions, let me know.